So we're talking about two-trait Punnett squares in this particular video. We're going to assume a Mendelian model of inheritance, which means that we're going to assume for the time being that the two genes are on separate chromosome pairs, something we call not being linked. We're going to talk about linked gene inheritance in a future video. So what if we had a doubly heterozygous parent, big B, little big T, little t? Then their DNA might look like this. In interphase, the DNA gets copied. They might line up like this in meiosis 1 to be split and to make gametes. And if you imagine that each one of those columns is a gamete, then you could have the big B with the big T and the little b with the little t in gametes. However, if another germline cell were to undergo meiosis, maybe they'd line up like this in meiosis 1. So if they lined up like that, you'd actually, when you split them up, you'd have the big B with the little t and the little b with the big T. So that parent could really make all four of those different gametes. And so if we were to cross that parent with a doubly homozygous recessive parent, then if we imagine that parent's set of genes and chromosomes, they might look like this. And really there's only one way you can line those up. So even if we were to copy and line them up in meiosis 1 like so, independent assortment could flip each chromosome pair around in different germline cells, but that really wouldn't make a difference. When we split them up and make gametes, there's really only one type of gamete that this parent can make, and it's little b, little t. So it's really important for a two-trait Punnett square problem to first figure out what are the gametes my parents can make, because if I were to finish this cross, then I'd put my two parents on the top and the side. I would show all the gametes that my doubly heterozygous parent could make, and just the one gamete that my doubly homozygous recessive parent can make. And so what that does for me is it shows me how big of a Punnett diagram I need to make. I only need to make a 1x4 here. So I'm going to build that, and I'm going to show those two gametes combining. Students sometimes when they combine these often write each parent's gametes kind of separate, and so they separate the B's and the T's. I would highly recommend that you write them together so that you can easily figure out what the phenotype is for each trait. I would put the big letters first and the small letters second. So this next one would be big B, little t, little t. And we can go further. Little b, big T, little t. And then on and on. So what do we have here? We could think about the phenotype ratio for this particular set. This is actually a very famous cross that we're going to talk about later. If we cross these organisms, then what we do with phenotype ratios for two-trait Punnett squares is sometimes we might have, let me get this going here, we might have up to 4 different phenotypes to consider here. And what we usually do is we consider how many of the organisms have both dominant phenotypes in the first box. So this organism would have both dominant phenotypes. The big B and the big T dominate. But I don't see that true of any of the other organisms, so I'm going to put a 1 there. The second blank would be dominant for one phenotype, let's say the B trait, but recessive for the other phenotype, so little t's would show the recessive phenotype here. That organism would show that. The next blank would be recessive, let's say now for the B gene, so they're recessive for that, but dominant for the T, so there's one organism who shows that. And then the last blank is for organisms that are doubly recessive in phenotype for both traits, and so that organism would be that. This is actually a 1-to-1-to-1-to-1 phenotype ratio. That means 4 possible organisms, so that basically means that 1/4 or 25% of the organisms should show each of the 4 phenotypic possibilities. When you cross a doubly homozygous recessive organism with a doubly heterozygous organism, this is kind of an important ratio that we will discuss more in the future. Okay, remember, to do two-trait Punnett square problems, you think about the gametes first. The two rules I think of is that the gametes always need to have half of what you start with, and they need one of each letter, or sort of one of each chromosome. So let's practice that maybe skill first. If I had a parent whose initial genotype was this, then when making gametes, they need half of what they started with, 2 letters, and they need one of each. So it could be like a big G, big N, or it could be a little g, big N. Those are the two that I see for that parent. In terms of this parent, all I can see is big F, little r. For this parent, doubly heterozygous, that means they should be able to make 4. Big, big, little, big, big, little, and little, little. 4 different gametes. So make sure you're very comfortable with that skill, because that's going to be absolutely crucial to doing any of these problems correct. So let's do a sample problem now. Let's say that purple stem color is dominant over green. So let's go ahead and make a set of letters for that. Let's make big P, say, equal to purple. Let's make little p equal to green. P, you could use a different letter if you wanted to make it more obvious, the difference. Tall height is dominant over short, so let's use capital T for tall and little t for short. And I'm crossing 2 heterozygotes, so they are assuming heterozygous for both traits. So maybe big P, big T, little t would be both parents. So if we were to think about the cross there, if they are both doubly heterozygous, then that means their gametes, they could make all 4 different gametes. Big P, big T, little p, big T, big P, little t, little p, little t. So I need to make a 4x4 Punnett square here. So I'm going to write those 4 gametes on the top and on the sides. This one will take you a while. Feel free to fast forward if you need to. I wish I could fast forward, but I can't. And I'm going to make a 4x4, and I'm going to try and do this on my tablet. Oh gosh. Probably should have created a square for myself through the program. I'm going to really suffer down there. Okay, so, and then you're ready to combine all your possibilities. Keep your P's together, keep your T's together. Oops. Okay, and we're ready to think about our phenotype ratio then. The phenotype ratio we're interested in, this is a very famous cross as well. I'll do the phenotype ratio up here. We're actually going to have 4 different phenotype possibilities for this Punnett square as well. We're interested in those that are doubly dominant for both phenotypes. Purple and tall, 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 purple and tall. That's my 6th one. 7, 8, 9. I actually circled this one. I should not have. I apologize. That one is actually green and tall. The 9th one is down here. So I apologize. There were 9 who were doubly dominant for both. There should be 3 who are green and tall. Here's green and tall. This one should have been green and tall. And this one over here is also green and tall. So there's 3 who are that way. I'll do triangles for those who are purple and short. Purple, short, purple, short, purple, short. And then there is 1 who is recessive for both. When you cross 2 organisms who are doubly heterozygous for both traits, then you should get a phenotype ratio of 9 to 3 to 3 to 1 in the next generation. 9/16 should be doubly dominant, 3/16 dominant and recessive, 3/16 recessive and dominant, 1/16 recessive for both phenotypes. That's a pretty famous ratio and one you should definitely keep in mind for our curriculum. Alright, let's do an easier problem, one that shows you that the Punnett squares don't have to be as long as the one I just showed you. If red eyes are dominant over black eyes, so I'll do big R for red and little r for black. And normal wings are dominant over vestigial wings. Then let's cross a fly who is homozygous recessive for eyes, so little r, and homozygous dominant for wings, and a fly who is doubly homozygous recessive. So those are my two genotypes. And crossing those, let's go ahead and rewrite the parent genotypes first. First goal of any two-trait problem is to think of your gametes, so one of each. That's the only gamete I can think of for that one parent. This other parent can only make this gamete. So, what do I really need to do? I just need to make a 1 by 1. Let's show the gamete that that one parent can make, the gamete that the other parent can make, and so all of the offspring will look like this. No need to write it 15 more times and make 4 by 4 for every two-trait Punnett square. Why not just figure out what gametes you can make and show how the different ones might combine? 100% of my offspring will show that genotype, and so they will all look black eyes and normal wings.